So welcome everyone to this evening's BMS talk. I'm going to start by introducing our chair for this evening, and it's Matt Wainhouse. Matt's a passionate fungal ecologist and conservationist. His PhD research at Cardiff University focused on heart rot fungi in beech and oak. He now works for Natural England as their fungi and lichen senior specialist. So thank you very much, Matt. I'll leave it to you now.
And thank you very much, Sally, and thank you, BMS, for the invitation to chair this really exciting seminar this evening. It's a great pleasure to introduce Christian Taylor, our speaker this evening, who's going to be talking on the role of wood decay fungi in biodiversity within forest ecosystems. Now, Christian has did his PhD on insects and fungi in rotting wood. That was his thesis with Alan Rayner at the University of Bath all the way back in 2001. I was just doing my GCSEs. But since then, since finishing that project, he's worked very squarely in the education sector. He's been an ecology teacher in various settings, as a biology teacher on farms, and in secondary schools. He's an associate lecturer for the Open University. He's a member of the Fungal Education and Outreach Committee and chairs the Education Working Group. And in his latest venture, he's running a transdisciplinary ecology summer school in rural France called the Rural Arts in Science and Education. And I will post a link to this in the chat for anyone to go and have a look, because it looks absolutely fantastic, really, really exciting. So with that introduction done and dusted, I would say, any questions that you've got, please add them as they come into your heads, post them in the chat. I'll be picking them up, and we'll bring them back to Christian at the end of his talk for a discussion on the role of wood decay fungi in biodiversity within forest ecosystems. Christian, over to you.
Thank you so much, Matt. Can you all hear me okay? I hope so.
Perfect.
Great. Yes. Well, it's wonderful to be here tonight, and thank you so much. And just a quick, just a small point of information. I'm not actually the chair of the education outreach group of in the committee of the British Mycological Society, but I am a member of that group. And yes, in fact, Mark Ramsdale chairs that group, and he's at Exeter University, and he may be here listening tonight. So if you are, hello. Hello, Mark. So yes, I'm here to really tell you a story about how fungi, often overlooked, as they often are, have another overlooked role in ecosystems, or at least often overlooked role. And this is really to do with the accommodation of other species in forests. I worked on the relationships between fungi and insects, but it occurs to me that actually some of the roles that fungi have in creating niche space for and with alongside insects, but also very much then later occupied by different successional waves of insects, that actually some ideas might be relevant to other animal groups as well. So that's just an idea that that leads to questions and further research ideas about biodiversity accommodation in forests. So we can talk about that a little bit more, perhaps some questions afterwards. But yes, so what got me interested really in this field, I suppose I had an attraction to it from a very early age, because this is a picture of the tree, the Spike Milligan tree in Kensington Gardens. And when I was very young, I used to go and admire all the nooks and crannies of this wonderful sculpture, which I think Spike Milligan actually restored, or he paid to have it restored. So I think initially it was carved by a wood artist and placed by the entrance to the playground. And I became rather intrigued, and it just sort of stayed with me. And it occurred to me when I was putting this talk together that that it might have somehow played a role. But really, when you look at dead wood in a forest, the most important thing to realize perhaps is it's not really dead. It's occupied, completely occupied by fungi, of course, being the only organisms that can really decay it completely and reconstitute the chemical nature of lignin into brackets and other parts of the mycelial structures and even fruiting bodies. Of course, the brackets are woody fruiting bodies. They have a kind of fungal lignin that's a reconstituted lignin from the breakdown of lignin elsewhere. And that breakdown of the lignin basically creates a kind of cavitation process. So I think the key point of this talk is this idea of cavitation within wood. So something that appears to us to be a very solid thing when it's intact, and of course within the living part of the tree when the tree is alive, gradually becomes full of, if you like, a kind of Swiss cheese effect. And this hugely magnifies the surface area, but also allows larger and larger groups of organisms to become accommodated. So it's only fungi that really can do this. So fungi, from the very initial standing point, are unique in their ability to decompose the very complex nature of lignin and gain their energy from that. So the particular part of the life cycle of fungi which I'm focusing on in this talk is the mycelial part, and how it occupies, ramifies its way through dead wood. And I'm really wanting to talk about the emergence of niche space as an emergent process from the interactions of fungi in a community inside a piece of dead wood, as much as also the interactions that the fungi community have with the grazing insect community. And when I talk about grazing insect community, really we're talking about the larvae of many of the insects that dwell in a forest under bark. And of course, when we talk about insect life cycles, it's the larval stage of the insect life cycle. It's the predominant part and plays the major role in terms of at least grazing on the mycelium, but also the major role in terms of the lifespan of the insect. So, you know, when we see the winged insect, which we find easier to identify, actually it's coming from a larva which is pupated, and that larval phase is the key part in terms of this talk, and it's the key part in terms of where the insect spends most of its life, perhaps inside a piece of dead wood, very often grazing, either grazing directly on the mycelium of fungi, or actually ingesting fragments of wood and having a kind of fungal garden in the microbiome of the guts, the gut structure, the intestinal structure of the insect. So it's known that many insects carry fungal gardens in their intestines, and that's actually how they're able to obtain the energy from ingesting the wood. So if we're lucky enough to be wandering around in a forest, and if it's humid enough, we might lift a piece of bark and find a rather nice mycelium like this one, growing along and ingesting, digesting the lignin as it goes. So this is the principal part of the structure of the fungal organism which the insects are interfacing with in terms of insects within rotting wood. Now, of course, there's another whole talk that's possible. So a friend of mine, Roger Guevara, did work on fruiting bodies. So there are lots of insects that also live in fruiting bodies. So I'm not going to be talking so much about those in this talk. But yes, we must also acknowledge that, yes, there's another group of insects that do inhabit the dynamic spaces and inside the little pores of the fruiting bodies. Many of the bracket fungi have wonderful beetle species that live within their fruiting bodies, within the tiny structures of the pores and within the architecture of the bracket. But in this particular talk, I want to focus on the mycelium a bit more, and the dynamics underneath the bark and in the wood itself. So to do that, we need to think about what wood looks like under the microscope, and consider how fungi actually get into the wood and ramify through so successfully. So one of the reasons is because xylem tissue actually consists of tubes, xylem tubes, which I'm sure many of you are familiar with. And so when we look at them in cross-section and longitudinal section, we can see these tubes under the microscope very, very easily. But also just to consider that fungal hyphae is superbly adapted to fit through those tubes and enter into this very high surface area world of tubes, which they can then digest as well. So this cavitation process really begins with the entrance of fungal hyphae into wood, and then the gradual digestion of these lignin rings, and in so doing, the first cavitation process we can imagine that starting quite early on. But of course, there's some classic examples that we should also be aware of, which are well documented, of insect species that also inoculate fungi into wood. And so ambrosia beetles are a very good example, actually both inoculating and cultivating mycelium as their food source within the breeding galleries that they create. And they have special, superbly adapted features on their carapace and their head to carry fragments of mycelium. Indeed, so under the microscope one can see these features as well of the ambrosia beetles. And here we can see the gallery, the classic sort of ambrosia beetle gallery pattern. So if you see that, you know you've got something like an ambrosia beetle, although they're different species of them. And of course you can see that what's nice about this, you can see that the tunnels that the larvae create start off very small, because the larvae are very small when they first hatch from the eggs, and then as they progressively chomp through their meal, their mycelium in their mycelial galleries, they get larger. So the tunnels actually have to get larger to accommodate the larger body size of these larvae, which explains why you see this slightly widening shape. But what's also interesting about that is that getting larger as an insect might present a problem. If your mycelium is found in the finest crevasses and microfissures of wood, then if you get if you're an expanding body size, you might then get subsequently excluded from your food source. So I'm going to talk to you in a moment about a group of insects that I looked at that has a very neat solution to that, which is quite unusual and interesting, but more in a minute on that. But first of all, a couple more examples of sort of classic examples of interactions between insects and fungi. So of course, we must mention the famous leafcutter ants, because it'd be wrong not to mention that. So here the mycelium is being grown and cultivated by the ants by feeding the mycelium the leaves, the fragments of leaves, and then the mycelium is responding to the diet in different ways and giving signals back, chemical signals back to the ants to tell the ants when to search for different sorts of leaves from different trees. If the chemical profile of the leaves is too unpleasant, slightly toxic, then the ants are told to hunt for another tree. So that's some wonderful biology going on there with leafcutter ants. And of course, the same kind of interaction exists with termites. Not all termites, but some termite species do a very similar thing and have a fungal partnership and cultivate a fungus with all the wood chips that they carry down to the fungal galleries. And also termites are also quite famous because in their guts, they also have these symbiotic fungi that we mentioned earlier in their sort of microbiome that help them to digest wood as well. So they use fungi to, if you like, you can consider different species of termites as having different sorts of dependence on either external fungi that are in their galleries, if you like the leafcutter ants, so more like those, and other termites that have more of an internal fungal garden, if you like. So quite an interesting group of organisms to think about. So let's move on to the work that I'd like to present about this very peculiar group of insects that does something very interesting and novel in regard to the problem presented by an expanding body size. So first of all, where do we find them? Well, first of all, in hazel woodlands, you can see here I'm pointing at a fruiting body of an ascomycete fungus called Hypoxylon fuscum. It's got hard, sort of rather dry feel to it, and it's there persistently throughout the year, but only fruits probably not all the time, but it actually, violently discharges its spores like the ascomycetes do. And then you can just about make out on the very top of the picture, there's a bracket fungus there as well. So we've actually got more than one fungus inside the same piece of wood. And that's the first thing to say, is the group of insects that I looked at was tending to be found only when there's a community of more than one fungus. And not just that, it had to be fungi that were very different from each other taxonomically. So this is a good example because it's a hazel log and it's got a basidiomycete fungus on the top, bracket fungus, spore dropper, and it's got the ascomycete fungus where I'm pointing the dark one, which is a spore shooter. So when we of course, we can in the lab, and this is what I did, cultivate the fungi in a kind of monoculture environment. So we can take both of those fungi and grow them separately in petri dishes on 2% malta extract agar. And that's a little bit of a process because going from wild fungi into the lab, it takes a little while to subculture and then eventually get your nice lawn, your nice particular well, this is halfway through that process actually, so it's still sort of suffering from a bit of bacterial contamination, but eventually you get a nice culturable fungus from the wild, and one can do that with both partners. So you can do that with the ascomycete and the basidiomycete. And you can also then introduce the insects. So this is a picture, a kind of mugshot top left of the head of a cecid larvae, a cecid larva. So cecids are a group of flies, so in the dipteran group, which are obligate mycovores. So they only eat mycelia, and that's their primary food source. And they have a tiny little mouth part which is superbly adapted to sort of strafe along individual hyphal threads and start sucking out the protoplasm. So they are really very well adapted for grazing on mycelium. And the other really interesting thing about it, well, they have a couple of sensory pulps you can see on each side of the head, and a few other smaller sensory pulps just around the mouth part. But the really interesting thing about them is that you can just make out on the actual coloured part of the image, there's a mother, and I'm calling her a mother now because in fact they're all female, and inside her are some tiny little larval babies ready to be born in a birthing process called paedogenic birth, in which the mother bursts open from head to tail, and out come her clonally asexually produced offspring. Up to 12 of those are produced at each paedogenic birth, which is a remarkable way of reproducing, and it means that they're entirely reproducing within the female lineage, and also they're not going through a pupation and producing adults. So this is what Darwin referred to as a kind of contracted life cycle, a bit like the axolotl, in fact. He talks about it a little bit because it was discovered in Russia that these insects did this right about the time when Darwin was writing Origin of Species. So it created a bit of a stir at the time, but since then very little work has been done on this group of insects. But my role was to try and find out a little bit about the ecology of these insects, and by doing so, perhaps explain why they might have chosen such a bizarre way to reproduce. And yeah, the interesting, you can also see bottom, there's a slightly more faint orange larva underneath the agar. So the middle one is grazing on the surface mycelium, and then we've got one underneath grazing on the mycelium that is buried growing through the agar jelly in a petri dish. And yeah, what's also very interesting is that they have ovaries within the larval stage. So most insect larvae do not have any sort of reproductive organs ready. They wait until they've pupated and they're in that adult winged alate form. So this group of insects is really unusual, having ovaries, but the ovaries produce eggs that don't need to be fertilized, and so develop spontaneously into these daughter larvae that then burst out at a certain stage. So the other thing about looking for these in the wild is that when I was looking inside the pieces of wood where the larvae were, I found that there were these black lines, and partly because my Alan Rayner, my PhD supervisor, had done some work on pseudoscrotal plates, I knew a little bit about these. And so it occurred to me that maybe there was something about these these black lines that might have something to do with finding these or locating the populations of these insects. So I started to hypothesize that perhaps the insects might find some attraction to residing near these pseudoscrotal plates. But yeah, it is actually interesting. So if you ever want to know, is there more than one fungus in a piece of wood, you just have to break the piece of wood or cut through it and look for these lines. And the lines demarcate the territories of one particular mycelium of one species, and on the other side of the line, you'll have a different mycelium from a different species. And because they're taxonomically different, they basically build this kind of skin between themselves, a kind of wall, which helps to basically partition the resource and avoid out competing each other. So effectively, it's a way of dividing the cake and saying, you know, here's my bit, and that's your bit, and a kind of territorial marking. But so that's a kind of neat thing that you can do with any piece of wood. Even small twigs sometimes will have a community of different fungi within them. Carpenters for many years have actually called these black lines spalting. And it's been something that carpenters have used to create beautiful bowls and furniture. But actually, it's produced chemically by a melanisation process that involves carbon compounds being adhered to one another. And by doing so, it mops up free radicals, which are otherwise very hostile to either of the mycelia on each side. So mycelia very often create quite hostile free radical chemistry in their pure cultures. And so, one thing I looked at was whether or not the PSP's lines had a reduced amount of free radicals and so on. And indeed, that was in fact the case. But the other interesting thing about the PSP lines or pseudoscrotal plate is that it can be regarded as a kind of carbon store, because it is effectively a very durable, a permanent feature in the wood that even persists after the rest of the wood has completely disappeared and decayed. So you get this kind of fungal char, if you like, or a fungal charcoal. It's not obviously charcoal, but it's produced by fungi, and it's a carbon compound. And there must be a lot of this in forests that's very much overlooked. I mean, I haven't actually tried to estimate how much carbon there would be stored in a forest, but it would be a really interesting thing to try to estimate. So here's some nice examples of pseudoscrotal plate being adopted by carpenters to make lovely bowls and so on, and that's nice. Yes, the thing is, perhaps not all carpenters do realise that in fact these are created by individual mycelia. So here, back to the woodland, back to our hazel log. If we take the bark off, on this particular occasion, I was lucky enough in my method of destructive sampling, which was what it's technically called. Basically, I was taking the bark off rather delicately, many pieces of wood in a wood pile left by some people who'd been doing coppicing in the hazel woodland where I was near Bath at the time. And I noticed that there were these two rather large cecids sitting on a pseudoscrotal plate line between Hymenocete corrugata at the HC and Hypoxylon fuscum. So again, it's a basidiomycete, basically on one side of the PSP line, and on the other side is an ascomycete. So taxonomically, really different groups. So that's the photo that sort of got me really interested in exploring this further. Here is actually a piece of PSP that's quite thick, and there's little crevasses inside, and these shapes that look a bit like bananas are actually dried hemipupae, which are when conditions get very dry in the summer, I was finding that the larvae dried out, and what's been shown in previous research is that these hemipupae have the capacity to rehydrate, and the little dormant larvae that are already inside can then emerge and start the life cycle again. So that's a really amazing feature of the biology of these insects. So far in terms of the exploration, in terms of this talk, we've got a situation where there are fungivorous paedogenic larvae of a particular species that I ended up identifying, called Britannia fraxinicola, so a particular cecid species. And it lives under the bark of decaying hazel, Corylus avellana. And we found these insects inhabiting interfaces between in principle, two groups, the Basidiomycetes and the Ascomycetes, but exemplified mostly by Hypoxylon fuscum interacting with Volumnia comedens or Hymenocete corrugata. But in this case, it was in the mapping I was doing, it seemed that Volumnia was more often associated with the PSP. So we went for that one in the end. And that the insects specifically inhabit narrow subcortical niches of dead wood between adjacent fungal decay columns, which contain zones of melanized pseudoscrotal plates. So we asked ourselves, do cecids influence the interaction between the fungi? If so, how much and how does that work? And is there reciprocal? Do fungal interactions in turn cause any changes in the insect behavior and the growth of the insects? So I'm going to present the results of some of that work, which I'm hoping to actually hopefully publish at some point soon as well. So I'm working on that. So here's the woodland site. You can just see it top right near Bath in Conkwell, near Dundas Aqueduct, just outside Bath. And bottom left, we can see Hypoxylon fuscum. Volumnia comedens is a Basidiomycete that actually causes bark to roll. And so you can see this sort of white crust with the rolled bark there in the second image at the top there, on the bottom right panel. And then we can see actually quite a few cecids in the bark layer. Just a reminder that they don't form any adult winged stages, or very, very rarely, only under extremely stressful conditions do they revert to forming any winged adults. And the only way to identify the species, which is what I mentioned, I had quite a lot of work to do to identify the species we found in the field, is you have to take electron microscope images of the tiny patterns on the head and actually the different surface features of the skin of the insect larva, and then compare those with known cecid patternings. So each cecid species seems to have slightly different patterns, and they're called welts. There's an interesting term for them. So it's a bit like a fingerprinting exercise. It's only by doing that work that you can actually know which species of cecid you're working with. So in this case, it was Britannia fraxinicola. So here we have our combined culture. So this is where we can grow the Hypoxylon fuscum at the top, the slightly darker looking mycelium, with the two inoculation holes, or actually taken from a previous culture. So the two circles represent where the mycelium was introduced into the petri dish. And there is actually a third one in the white area down below, which is the Volumnia comedens mycelium of the Basidiomycete fungus. And you can actually see on this image quite a lot of these cecids clustered, it seems, along this boundary, this PSP line, even in culture. So that was interesting. But also, you can see that they are going away from that PSP zone. You can see some little lines, for example, if you look closely on this image. I don't know whether you'll be able to see it, but you can just about make out some little lines at the bottom, and those are the trajectories of the insects as they graze through the mycelium. Now, one interesting thing is if you don't have any insects at all, if you remove the insects and just grow the interactive fungi, the Volumnia comedens invades the territory of the Hypoxylon fuscum over time. So even though the Hypoxylon fuscum is producing a really good PSP, and of course the Volumina comedens is also contributing to the production of that PSP, because it takes two parties to basically create a PSP zone. Even despite that, the Volumina seems to have the upper hand and seems to be able to invade the territory over time if you leave the culture growing for long enough, and eventually the Hypoxylon fuscum is wiped out. So with the insects present, that didn't seem to happen so much. So that was a first interesting observation. So just very briefly, if I just point out at the bottom picture where the bar chart is, you can see that we've got cecid, the actual density, so the number of cecids per centimetre squared of Petri dish. And you can see there that there's definitely a much higher population found close to the PSP or on the PSP zone than on either of the two either of the two mycelia on either side. So they do seem to be clustering on the PSP zone. The other most interesting part of this image is the fact that we can see, if you look at C, there's an image with 3 Petri dishes in a row, and it's basically time a kind of time-lapse snapshot. So with early cultures, you can just see the PSP beginning to form. And then over time, you can see that these PSP zones do get much more extensive and thicker. What's also interesting is that if you look at the graph with the spikes, the spiky graph at the bottom right, there are particular features that are arrowed, particular chemicals. This is high pressure liquid chromatography. So basically it's running the extracts of the mycelia and the PSP through different columns to do this chromatography work to see what the chemical profile is of these different exudates are from the mycelia. And arrowed are unique profiles of the PSP. So the PSP is producing its own unique chemistry, in other words, which is not shared by the chemistry of the pure mycelia on each side. So one of the experiments I did was to look at this, what happens if you add the insects, what happens if there's no insects, and the top image with the blue and the red trace show the control versus with cecids added, how much the PSP sort of grew in thickness. So effectively, what we can see there, just visually there, is the size of the thickness of the PSP is much enhanced with the interactive presence of the insects. And that's confirmed by the data on the bar chart, where you can see the PSP area in the dark blue bars. You can see where it says PSP, it's much higher than where we have the cecids present. Where you have the light blue, there are no cecids. So effectively, we have a much thinner PSP, and we have consequently larger territories of the pure mycelia. So the insects do seem to be having a role in shaping the PSP zone. Also, what's nice is to measure the radius of the trajectories of the insects. And the reason I did this was because there was some work going on at the university where I was studying on ants and ant foraging loops, foraging trajectories. There's some theory about foraging behavior of animals, which is that if animals are walking in little circles, quite tight circles, it means they're basically quite fond of the area they're in. But if they're going in straight lines, the theory is a very oversimplification of the theory, but basically if they're walking in straight lines, they're trying to get out of a hostile area. So what we can see, if we follow that theory here, is that close to the PSP area, we have tighter turning circles, and further away from the PSP, we've got the straighter lines. And this suggests that the insects are quite fond of staying close to the PSP boundaries, which is what we've been observing in the field. So some other work we were able to do in the laboratory was to film the insects with a time-lapse camera, basically looking at how fast they're moving in culture. And so what we find here is that the newborn larvae tend to be moving the fastest on the interactive cultures, but actually they're moving a little bit slower when it comes to non-interactive cultures. So we can see here on the left-hand side of the bottom left bar chart, we can see the mother actually moving quite slowly on pure culture, and the newborns are moving considerably faster in non-interactive. That's just pure hypoxylon fuscum. Whereas when we grow the interactive cultures and put the cecids in, we can see that they reach a faster speed, but the mothers seemingly don't. The mothers, if anything, have slowed down, which is quite interesting. But so there's some speed differences, which is interesting. In the middle of this figure, you can see the data on the turning circles that I just referred to in the previous slide. So the basically we've got our turning circle radius measured vertically on the x-axis, and then we've got the PSP in the middle there with the smaller turning circles, and then so you can see that data there. And then on the right hand side we can see two bar charts there. The top right, we've got the body size. So this is the length the insect reaches before paedogenic birth, before basically splitting open from head to tail and all the daughters wriggling out. And what's interesting is that in the interactive cultures between Hypoxylon fuscum and Volumina comedens, they reach a larger average size before giving birth. So they're giving birth perhaps at an earlier stage, or choosing to do so, or the conditions are such that, if you like, they're not growing to that full extent, giving birth at a smaller stage in the pure cultures. And also the final graph at the bottom right shows that cecid populations are growing faster when you've got the two different species. So when they've got you know, there are different ideas about why this might be the case, but basically they're reaching a higher population more quickly on the interactive cultures. So here's the actual raw data at the top on the cecid length measurements for the interactive cultures. So in the red, we've got the monoculture, so if you like, just growing on one fungus or another, and then we've got the interactive cultures, and you can see this slightly elongated body length before birth, and you can see that data summarized below as well. And so what does all this mean if we can sort of put it all together? Well, going back into the field, armed with the information from the lab, what I tried to do was put together a kind of overall picture of what might be happening, and explaining also this extraordinary life cycle of these insects. So basically, first of all, PSP lines are often associated with small cracks and fissures. And you can see this when you look at a piece of wood that's very, very decayed. You often see this kind of darkened surface. If you look inside some of the cavities, often it looks like there's been a fire, because what you're looking at is actually where you're looking at just the PSP remaining, if you like. But sometimes when you crack a piece of wood, you'll notice that it cracks along a PSP line. So what I did in the final experiment is I tried to look at actual growing the fungi interactively on pieces of wood in a controlled way, and then adding insects to different types of treatment. So I had just moist wood on its own with no fungal addition, and then I basically got the wood to grow Hypoxylon fuscum by incubating it sandwiched between petri dishes of Hypoxylon fuscum culture. So that's how I tried to get the wood to grow Hypoxylon fuscum by having thin enough slices to basically put in a petri dish and put another petri dish on the top, and so sandwich the wood together. So I sort of created these pieces of wood that were inoculated by the different treatments. So we've got a control, we've got Hypoxum fuscum ascomycete on its own, Villaminia comedens, and then we've got the key one is the interactive one, HF plus VC, so that's Hypoxum fuscum plus Villaminia comedens. And then we've got the key point is to add the cecids. So the same number of cecids were added to each of these logs, and I had, I think, 50 ice cream tubs full of these pieces of wood on little sticks, on little poles to hold them apart from one another. And then after 10 months, counted the total insect population on each piece of wood. And what we can see on the graph below there, bottom right, is that at least from this experiment, we can see that the numbers in total reach a much higher level. So mirroring what we found in culture in the wood as well. So we're finding higher populations in the interactive cultures. So putting it together a little bit more, when I looked at the pieces of wood, what was clear was that the PSP very often was associated with lifting bark, and that's a key aspect of the way that insects might be then finding little channels to explore, to graze for further mycelium. So perhaps it's about access to food. When you put all this together and then look at how wood decays with this interactive culture with the PSP, you can see things happen that explain in an architectural way how the fungi are contributing to creating the conditions whereby an architecture emerges that basically suits the insects in terms of accommodation. So in a sort of schematic view, I sort of penciled this together quite a while ago, but it still works. Basically, the red circles represent the different body sizes of the cecids. And as they get bigger, they get basically excluded from the smaller crevasses and smaller microfissures under the bark. But because the PSP zone is associated with bark lift, we then see that actually the smaller of these circles, the small daughters, can then wriggle quite nicely out of their mothers and into the food on either side of the PSP, into the microfissures which the mothers were becoming gradually excluded from. So there's this whole sort of dynamics going on, basically, which is really exciting and interesting about how a piece of wood eventually accommodates a whole community. And if we take this a little bit further, well, I'll do that in a minute. So first of all, I'll just quickly summarize where we are. It seems that paedogenic cecids, Britannia fuchsinicola, exploit narrow subcortical niches of dead hazel that's been colonized by the fungus Hypoxum fuscum Villaminia comedens. The insects specifically inhabit interaction zones containing the pseudoscrotal plate. The presence of cecids influences the interaction between fungi, causing an expansion of the PSP. And the fungal interactions in turn are causing changes in cecid behavior, as well as increased larval size and enhanced cecid population growth. So this fits the models of the theory of niche construction, but also what I'm going to add to that is niche emergence as well. Yeah, so basically, what does this all mean? Well, when you look at a piece of wood, this is a piece of ash that's, you can see lots and lots of PSP very often, where there's been an extensive community of many fungi over many years. And some people have even said that PSP, because it stays put after the wood decays on each side, actually creates this kind of manifold surface area structure, which then can accommodate so many other insects in sort of waves of succession of gradually increasing body size. So when we think about how that might work with forest succession, basically, in terms of theory, we could add the idea of cavity number and cavity size distribution within the idea of a classical sort of forest succession. So that's a nice potential addition in terms of what fungi are doing. And of course, we need to also think now about hollowing processes. So of course, fungi are well known for creating hollow trees. And so we often find a lot of PSP inside hollow trees. So putting a schematic together for that, it seems like there's a sort of sweet spot between the death of a tree or the dysfunctionality of when a tree no longer is actually actively alive, and therefore this occupancy can begin. And then the gradual decay and the increasing fragility of the structure that eventually falls apart and becomes soil eventually for the next cycle. So somewhere in the middle where the blue line is, we've got this magical sweet spot where we've got this maximal cavitation holding together this manifold structure for the accommodation, or providing accommodation for many, many different insect species. And here is just a picture of many insects that I trapped from I made a series of these little wraparound insect traps, which you can do quite easily with mosquito netting, with a little pot to collect the insects at the top. And basically it's called an emergence trap. And so on different fungal occupied logs in the Haber's coppice, I was able to create these traps and basically look at the diversity of insects emerging from just one little short section of wood. And it was very, very interesting just how many there were, how many different groups of beetles and even small wasps, many flies. So just towards the end of the talk, what I want to do is just give a couple of other examples as well of maybe where this could be going for future work, for anyone wanting to look for an interesting research project to take this kind of thing forward. So here is a picture I took of a stag beetle larva. It's black and white, unfortunately, but you can just see the head above the number 5 poking out of a crevasse in a piece of oak, which I've lifted the bark from. And it's the same one in the top right image. What's interesting is that this crevasse is actually on a PSP zone. We can see the white is a Paedogenetic fungus, and then we can see that this dark area around, which is most probably well, we know for sure it's a different fungus, but just to suggest that there may be links with other groups of insects, really, this image. Also, rather circumstantially, the only place in the UK where you can find the violet click beetle, which is a lovely beetle, is actually within very, very large veteran oak trees, where if you look inside the hollows of them, nearly all of them are hollow, you find large amounts of PSP that's been accumulated over many, many years. And we mustn't forget that, of course, many of the creatures we love the most in woodlands live for a substantial part of their life cycle inside hollows created by fungi. And so whether it's bats or owls or squirrels or dormice, and one could also, I suppose, think that the act of making a bird box is really replacing what the fungi are doing in larger diameter pieces of wood over time. So appreciating the fungal significance of biodiversity accommodation, I think, is something that could be done. And this is just a little story to round off the talk with. I was very lucky to do some time in my degree on placement in Canada, working in a national park there. And I came across this woodpecker species, which was beautiful, called the pileated woodpecker. And what's interesting is people told me that the courtship ritual of this woodpecker involves the male very rapidly tapping its beak on a sapling of a pine tree, and the speed of tapping seems to be interesting to the female. So they tap very vigorously, but what's been later discovered is that they actually are inoculating through their beaks a fungus into the saplings of young pine trees, and then that sapling basically it grows latently, it appears, because later that tree basically becomes a large tree, and generations on from the courtship ritual taking place, there's a tree with the right fungus for the woodpecker to excavate its cavity. And so nest sites of pileated woodpeckers are effectively, if you like, created by the conditions created by the fungal community inside the wood, and making it soft enough for the excavation process. But what's also really interesting about this story, because we haven't quite ended it, is that the pileated woodpecker, after several seasons, gets evicted by ticks and lice and that sort of thing. So they get a lower rate of the high mortality rate, if you like, of their young. So they have to go off to find excavate a new site. And inside the old pileated woodpecker nests, one of Canada's most important keystone species then moves in, the flying squirrel. So flying squirrels depend on the vacancy of these pileated woodpecker cavities. So the flying squirrel, which is this part, Eastern Canada's flagship conservation species, very charismatic and cute, and they take them into schools. But actually, really, perhaps the wood decay fungi could be called the keystone species that are creating the conditions, not just for the flying squirrel, but also the pileated woodpecker. So that's basically rounding off a little bit where we are in terms of perhaps the under-acknowledged or slightly overlooked role that fungi often play. But I'd like to just, before I finish, and we had some questions, just flag up that there is going to be a really wonderful event online for anyone listening who'd like to take part soon, around the October the 5th date of UK Fungus Day. This year, there is the British Mycological Society is going to also be running another wonderful international symposium, fungi around the world. And so if you come to the British Mycological Society, I think the dates of this have now been confirmed. So it's going to be a little bit after October the 5th, and there'll be this separate international event where researchers will be able to present their findings. So if you've been interested by what I've been talking about tonight, I'd like to hear more about fungal ecology and other aspects of fungal biology, including medical fungi, then do log in and take part in the symposium, which would be good. And just if you're interested in any references to do with what I've been talking about tonight, there's a couple here which will get you started. So if you're interested in the insects, Wyatt 1967 paper on pupal paedogenesis in the cecids, really interesting article, and about the niche construction idea and niche emergence, which I've been referring to in terms of the fungal role tonight. Well, there's this paper here. Well, it's actually a book, in fact, it's a chapter of a book, but niche construction, the neglected process in evolution. Okay, so I think that's pretty much the end. I'd just like to finally acknowledge all the help I've had to get here, as in being lucky enough initially to be invited to do the PhD and to having Professor Stuart Reynolds as the entomology supervisor, and Alan Rayner, my fungal supervisor, which was absolutely wonderful to have both of those supervisors. Dr. Roger Guevara's work, he worked alongside me on insects and fungi, looking at beetles that live in fruiting bodies, particularly bracket fungi, so do look him up. He works at INACOL now in Mexico, which is the Institute of Ecology in Mexico. The postgraduate funding I received from NERC, so Natural Environment Research Council, so acknowledging that, and also the fact that I'm very lucky to be on this education and outreach committee for the British Mycological Society. So yes, and also I am working at the moment to try to publish my papers from this work. So I'm still in the process of doing that. It's been a long time coming, so I'm hoping to do that now luckily, the work is still pretty current, and no one's done, as far as I know, anything quite along this line. So I'm very lucky in that respect. So I'm hoping to get published soon. And thank you so very much for listening. And yeah, that's the end of my talk. Thank you.
Thank you very much, Christian. What an absolutely fascinating talk. Just mind-blowing on every aspect. You're being showered with praise in the chat as well for very good reason. So I guess over to questions. If anyone wants to drop questions into the chat, there's some that have been already put there. I've made a note of those. Some have been answered along the way. But I guess I'll start with just coming back to the pseudosclerotial plates. And you were looking at the interactions there between a lot of the time a Paedogenetic and an Ascomycete. Is that, yeah?
Yeah.
And I wonder whether you could just give a little discussion on the roles of the two, the Ascos and the Paedogenes, and yeah, the different roles they have in creating those PSPs.
Yes, thank you. That's a really good question. And yes, I mean, it seems that the Ascomycetes have a chemical versatility, which means that they are able to produce lots of antioxidants, which help to create the PSP. So Hypoxin fuscum, at least on the face of it, seems to be doing a lot of the work to create the initial part of the PSP. But also the Paedogenetic have this kind of mycelial dexterity to somehow overcome that and sometimes leap over the top with their kind of abilities to create, you know, rhizomorphs and things. So but in so doing, then increase the amount of the free radicals that are actually floating around on the Ascomycete side. Now, those immediately then get sequestered into the carbon chemistry of the melanisation process that creates a thicker PSP. So actually, both are involved pretty much equally in creating the PSP. At least that's my feeling. Although, in fact, there is a research question there, as there is with so much about you know, fungal biology, because as I said, it seems like the process really starts with the Ascomycete, but then when it interacts with the Paedogenetic, it really seems to accelerate.
And then there's a couple of questions on the cecid biology as well, which I mean, I've never heard of these things, but I mean, fascinating things really. So one of the questions was about, do you think the cecids contribute to the dispersal of the fungal species?
Oh, that's a really, really interesting question. And it's not one that I looked at because, I mean, it's possible that some of the welts, these surface features that they have, which some of them are actually looking a bit like hooks, may play a role similar to the carapace of the ambrosia beetles in the sense that when they can wriggle along, they might locomote some fragments of mycelium along. So yeah, they're unlikely to be involved in spore dispersal because they are principally involved with the mycelium and potentially quite a long way from the fruiting bodies. So unlikely to be showered in spores to transport the spores. But yes, certainly could possibly be involved in the transfer of fragments of mycelium and the kind of, if you like, inoculation of mycelium into new territory. But yes, it's an interesting question, and the answer is really yes. It's another research question, really, how much that might be happening.
I get a feeling that a lot of this is going to end in. There's another research question there. You may have mentioned it, and maybe I missed it, but the cecids themselves, how do we know how they disperse?
Oh, yeah, that is an interesting question. So one of the things I noticed they did in culture is, I only noticed this in the Petri dish initially, is they can hook their heads onto their tails and flick themselves quite some distance. And I was quite surprised because I opened a Petri dish a couple of times and suddenly, bing, this cecid sprung out onto the lab bench, and I was thinking, wow, that's amazing. So then I tried measuring. So I actually did a preliminary, but it wasn't enough really to get much data, but I did spend about a week sort of measuring how far they could ping themselves. So it seems that they do have a way to locomote, which is a bit sort of random, but obviously can be effective in a coppice environment with potentially quite a lot of territory to explore. But also they can move like a maggot, like a larva can. So they undulate their way along as well.
Fascinating. And do we have any idea about how ubiquitous they are, how common they are? Are we finding them in all hazel coppices? Is it just coppices of certain age?
That's
or is this another research question?
I'm afraid, yes, it's another research question, but in an extensive coppice, I was finding them quite predictably when I knew what to look for. So looking for, when you're next looking in a hazel coppice, you might like to explore where you've got more than one type of fruiting body. So a community of fungi. Have a little look where there's any loose bark. You can peel the bark away quite easily, and you might find some, which would be great.
Yeah, it's definitely something to do. I was just thinking there about woodland connectivity, with the big drives to plant more woodlands, and it's kind of easy to sort of follow things like birds as they sort of migrate in, but something like this, you clearly have quite an important ecological function. How are they getting around, starting this cavitation process? It's a really interesting question.
It is. I mean, one of the things that's interesting is that very occasionally cecids do actually form a pupa under very stressful conditions and form a completely sterile adult. So that shows how it's only recently that these cecids potentially evolved this kind of new way of reproducing. Recently, I mean, still, we're talking, it could still be quite old enough for Darwin to have written about it, but recent in terms of evolutionary events, and recent in terms of this particular species, because when they're under real stress, they seem to revert back to a more conventional life cycle, but they lack the key component, which is the ability to actually reproduce in that adult stage. So yeah, this has made some people think about silence genes and different genetic mechanisms underlying all of this. So that's another whole aspect of the cecid biology, is they're very interesting genetics, because they have a very high ploidy number, up to 40 copies of each gene, rather than just diploidy that we have. So wow. Yeah.
Fascinating. So the next question, a couple more on the following on the cecids. One question was, are they exclusively paedogenic? It says pathogenic in the chat, but I think that's possibly meant to be paedogenic.
Yeah, so it's if you like, but parthenogenesis is a form of asexual reproduction, which yes does involve this bursting open, and this is the only way that they reproduce. You're quite right. Yeah, they're locked in.
So a question from Leah, which says, this is about the foraging speed, I think. So do the larvae grow? Do you think the larvae grew instead of the mothers, as they had to feed while the mothers were too large to stay close to the PSPs?
I think there is an architectural answer. It made sense looking back at the wood and thinking about these micro fissures, that yes, as the mothers were growing, they were getting blocked from the mycelium that they needed to feed on. So by, if you like, subdividing, which is effectively what they're doing when they do their paedogenic cycle, the mothers really are reconfiguring their body size and multiplying themselves at the same time. And it's a very, very ingenious solution to the problem of being excluded from your food by growing too big. So I hope that, I don't know whether that answers the question, but I hope it does. It's sort of somewhere along the lines, but yeah. I think there is an aspect of the explanation of the biology here, which is we have to sort of put together the architecture of the wood with the interactivity of the fungi to really understand the picture. What's going on? Yeah.
So a question about cecids in commercial growing now. This is from Roger Kemp. who says that they were plagued with these adult flies of cecids, phorids and sciarids, which were adults. So is this at odds with your work, or is it a different evolutionary path that these closely related species have gone down?
Yeah, so there are quite a few different species of insects and groups of flies that will plague mushroom cultivation, mushroom growers. So yes, you do find fungus gnats, for example, which are different from these cecids. And yes, it is something. In fact, the initial funding I had from NERC was to try to follow up some work from Justine Hunt, who'd done a PhD just before me on the insects that are growing in mushroom houses. But yeah, so I branched away from that a little bit just to look at the ecology of this particular species I was finding in the hazel coppice. But you're right, there's actually a group of fungus gnats that actually form adults quite routinely and will fly along and, as you say, plague the mushroom beds. But there are also cecids that plague mushroom beds that are true cecids that actually won't form adults, that just reproduce in this way and effectively eventually sort of totally infest the compost. So you have to sort of, I think, quite rigorously fumigate or clean out the mushroom house. Not being an expert on mushroom growing in that sense, but I believe that that's the case. So it's quite a process to get rid of these things once they get established.
A couple of comments that just came in as you were talking then. So fungus gnats are attracted to interaction zone lines in agar culture. And another comment that this strategy has evolved several times within the cecid lineage, concluding this because they are parthenogenic and there is some diversity within the group.
Yeah, that sounds very good comments. I mean, I agree with those. Yeah.
Fab. So next question is again talking about cecids. Are they found on decaying wood high up in the canopy, and if not, or in lower density, does that lead to a different decay architecture?
Wow, that's a really good question, and one I'd love to be involved with helping to answer by getting higher up in towards canopy. I mean, doing canopy biology is such an exciting thing to do regarding insects and also fungi, given that 70% of fungal activity is actually above your head in a forest, you know. So it would be so wonderful to get some funding, and if, you know, I mean, I'm too old for it now probably, but, you know, for anyone young to want to go in, you know, basically climb trees and get to that height and do that kind of work. So in other words, it's another research question, but it's a very likely to be the case, I think, that the cecids will be able to be in the canopy quite easily, where you have obviously slightly smaller girth of twigs and branches, but nevertheless, if there's a community of fungi up there, which there is, it's quite likely that we're going to have them quite high up as well.
So just to change tack then away from the cecids for a moment, and just thinking more broadly on the conservation of fungi or insects as well, really. I'm just wondering, me as a conservationist, you know, really, and trying to shoehorn fungus conservation into every conversation I have. Do you think we're missing a trick in not exploring this relationship a little bit more detailed, or at least making more of this relationship between fungi and invertebrates, or probably those big cavity using animals, the fluffy things with backbones?
Yeah.
And is that something we should be doing more of as mycologists?
I try to mention these kind of interactions when I do fungal forays in Devon, and people do seem to be really interested in it. And I think that the message is gradually changing some policies around land management. For example, I think the National Trust now leave dead wood a lot more. So that in terms of the message about dead wood being valuable, I think the message is being received. And to some degree implemented in terms of a change in policy and a change in what we're seeing in terms of the amount of deadwood now being left. I think standing deadwood is more valuable than horizontally lying down sort of log piles, and I think that's because potentially there's a greater heterogeneity of humidity in a standing dead tree as opposed to one that's fallen over. So that's going to reflect in fungal diversity again, with different humidity levels of different species tolerance ranges, and then potentially greater diversities of insects. So I don't think the message has quite got there yet. I think the message has been received about deadwood, but I think not enough people appreciate the value of a standing dead tree yet to see the beauty of it and see how valuable it could be in terms of generations of insects in that area, and therefore birds and other wildlife depending on it. So yeah, hopefully in time the message will get through, but I think that perhaps we could do more in the fungal community to help to get that message across by talking about the valuable role that fungi play in this.
Do you think we need to be, I don't know, interacting much more closely with entomologists, particularly, I guess, with the field mycologists, maybe? We are segregated little groups, I suppose, little islands of mycologists and entomologists. Do we need to be getting together to thrash out some of these questions?
That would be brilliant. I mean, in natural history society groups and so on, it would be lovely to get these conversations going, because I've had conversations with entomologists who believe that wood decay occurs because of insects. And so, of course, with the fungal lens, looking at wood decay, we see a different story, but it's the interactions that are so often the key, as is always the case. So I think interactions between entomology and mycology would be really good. Yeah.
You mentioned the role of invertebrates in wood decay then, and it's kind of a pertinent question as we're particularly as you're trying to understand carbon flows and carbon dynamics, but and maybe it's a pitched battle between mycologists and entomologists, I don't know, but who is making the biggest contribution to wood decomposition? Is it the fungi, or is it the invertebrates?
Well, it's again, by looking through fungal spectacles at what's going on, it seems it's clearly fungi that are actually doing the very basic stuff of allowing the first cavitation to take place, without which you wouldn't have bark lift, you wouldn't have any cavitation of the xylem tubes as they sort of merge into each other as the walls decay. So, but then again, without the insects moving in, of course, none of that material, well, some of the material would be removed, so that the insects definitely contribute. So you get successional waves of insects. And I would say the story we've told tonight about the cecids is just the thin end of the wedge. It's the first stage, really, of this successional sequence in which you get greater and greater cavity numbers and sizes. So yeah, it needs both. But fungi are definitely the starters of the process, I think. Yeah.
And obviously your research is temperate. We say that's the same for the obviously no termites here in the UK, but them being fairly prolific wood munchers. Do we think it's the same for the tropics?
Well, that's again another research question, but I'm actually going to Mexico in October to do a bit of field work with Roger Guevara, who's mentioned here that he's working at Icology, and we're going to go and look at pieces of dead wood, looking at PSP in the tropics. So I'll try and report back. But I believe the case. I believe so. I believe that it is something that's not just in the temperate forest, but we'll see how much we find out there.
Fantastic. There's another question in the chat, which is, how did you arrive at the 70% of fungal diversity in a forest being in the canopy?
Oh no, not the diversity, it's the activity. 70% of the activities of the wood decay is because well, I believe, I mean, it's I kind of got that from Alan Rayner really, but you know, I believe it's the case that for a large bit of timber to actually fall, in other words, for a branch to actually fall off, about 70%, at least 70% of the lignin has to have decayed for it to become, if you like, soft enough to be broken off in a storm and so on. So the actual chemical activity of decaying the lignin, 70% of that is above ground before anything actually reaches the forest floor.
And then I was leading on from this, and I wouldn't expect you to know the answer, but your thoughts maybe would be welcome, I guess. And again, this where we're starting to think a lot about carbon counting, particularly in the creation of new woodlands, and how much carbon we're going to sequester over that time. But if we think we're losing so much in the canopy, obviously some of that is being sequestered in the soil. Do you think we're perhaps missing something from these carbon models that isn't factoring in this decay that's happening in the canopy?
I think one thing we're missing is the actual amount of PSP that is a very long-term durable store of carbon in forests. And we're not factoring that in at the moment because it's well, it's just a niche area that hasn't yet had a role to had an input into those kind of equations. So I think if we were able to do some work, if it was possible to do work on quantifying PSP in both tropical and temperate forests, we might see a considerable contribution that fungi are therefore making in their diversity of interactions by creating PSP that actually would be effectively locking carbon up, and therefore it might allow climax or near climax forest, instead of being seen as decadent sort of carbon emitting systems, to be seen as sort of locking at least quite a lot of carbon up. So it would enable mature forests to be seen more as carbon store. But obviously the equations would have to be done, but I think that it would be a very interesting exercise to do that.
Yes. Another PSP question, which is, how much does the PSP vary between fungal species, and are there some that are more aggressive, or is it a similar process universally?
It's a really good question. There are some subtle differences, but it seems that the chemistry of the melanisation, which is the key part, is shared between many different interactions. When we had the Ascomycete, I was looking at the Hypoxin fuscum, whether it was with, you could choose a variety of different Basidiomycetes to pair it with, but fundamentally the PSP zone would end up being pretty chemically similar. And one of the reasons I could tell that was because the interactive cultures, I had them in a separate incubator, and when I opened the door of the incubator in the laboratory, there was a waft of licorice which hit my nostrils, which you didn't get with any of the its pure cultures. And it actually made me sort of think about going down a line of research onto what, why does it smell of licorice and what's going on there. But there was a unique, definitely there was this, it fits in with this idea, there's a unique chemical profile of the PSP, but it didn't matter whether it was which particular Basidiomycete you were pairing the hypoxylon with. It you've always got this interesting aroma. Now the question would be, if we had a different Ascomycete fungus, would we get a similar licorice smell? I don't know. That's a research question.
Yeah, I mean that's what I was going to ask, is whether exactly that was the licorice smell, the hypoxylon that you can smell.
Yeah.
But do you get what about the relationship between, because that's obviously a soft rot, and was it just white rots that you looked at as the Basidiomycete? Did you look at any brown rots?
No, it was mostly the I think it was white rot in principally, yes. So no, I didn't look at brown rot, mostly because in hazel poles of quite small diameter, there's we're not finding a lot of brown rot fungi. Brown rot fungi seem to be more in the larger diameter woody debris. So yeah, we did find a lot of these white rot fungi. You're right. So that's another, yes, another leading to another question, which is really lovely. That's what's nice about these question sessions, is that there's lots of ideas.
Yeah,
lovely.
I mean, the reason I ask about that was just looking in Oak and some of the work that we've been doing that we have done in Cardiff, is you just saw a lot fewer of pseudosclerotial plates. It was very difficult to actually work out where one fungus started and the other finished. You know, it was a lot more blurred.
With the brown rot, I see. Yes,
brown rots, but I wasn't sure whether you got that with whether you looked at that at all. But one of the comments that came in, or at least a question, was about the smell of the pseudosclerotial plates. Did you notice any distinct smells? I think you answered that one on the licorice.
Yeah.
And then there was a comment from that trees also shed branches without any decomposition. They just drop them because they don't need them anymore. And then another question on, do you assay for peroxidases?
Oh, that's a really good question. Yes, the answer is yes. I did some work with a method of tissue printing by taking a membrane, nitrocellulose membrane, with growing that with the fungus, and then being able to expose that membrane to different chemical tests. And so I looked at peroxidase activity either side of the PSP and also within the PSP, and was able to see bubbles of the being produced where the activity was most intense. So that was really an interesting bit of work, and I've got some figures in my thesis where you can see the different areas where the peroxidase activity is most intense. So it seems just either side of the PSP, you get this really intense amount of bubbling occurring and associated with the peroxidase. So that was also interesting. And then I looked also at some other techniques involving basic free radical, detecting free radicals using a similar technique, so presence of free radicals. So there was a really interesting band associated with where the peroxidase was active, where you saw very little free radical action, or very little presence of free radicals just near the PSP. So that sort of leads to this sort of chemical thesis or chemical hypothesis that the cecids might be just simply, you know, finding refuge amongst, you know, an otherwise quite hostile diet.
And just another question for me, I guess, on the experimental setup. I love the idea of you standing there with a speed gun looking at some of these cecids, but I imagine that's not the way. But how do you measure the speed of some of these things at such a finite, such a tiny scale?
Yes, well, it was really just done with the time-lapse imagery with a Bolex camera that was inverted over the interactive cultures with the cecids present, and basically just being able to look at the speed at which they were moving around the petri dish. So it was there was a process of looking at films really under time lapse and seeing, actually tracing the movement of the individual cecids, picking particular ones to follow. Not all of them could be looked at, but getting enough data to show something. Yeah, it was a really old camera, actually. I seem to remember that. I think these days it would be easier to do with a more up-to-date device.
Yeah, no,
yeah.
I imagine. Question from Joette. Hello, Joette. So Joette's saying, or asking, sorry, we observed significant temperature increase at the zone of interaction around contamination in grown mushroom substrates. Did you ever look at temperature changes around the PSPs?
No, I didn't, is the answer, and it would be really interesting to do. I got really interested, though, in the idea of temperature change being important for insects to overwinter. In other words, the effect of mycelial activity, basically raising the temperature of a piece of woody debris by a small amount, a really small amount, but enough to stop frost and ice crystals from killing the insects. And I think there's some really good work to be done to basically look at the role fungi play in creating very, very small temperature hotspots, and not exactly hot, but not freezing spots, shall we say, areas that are free from frost in forests, particularly in winters. And that would be a really interesting bit of work to do. And yeah, that's I think it would be easy to do now, because it's much easier to get hold of these thermocouples and put them on digital, so you can simultaneously take readings of temperature in very small places across quite a wide area. So that'd be really nice to capture that data digitally and look at these little temperature islands. But no, the simple answer is it would be really interesting to see if there is a temperature change associated with PSP. Fascinating, yeah.
Fab. And I guess we're getting close to our closing point. There's a quick question on the tunnels from the ambrosia beetles. So this was a question that was put right at the beginning. Are the tunnels made up of the larvae all coming out the same central point? Yes.
Yes. So the eggs are laid in the initial tunnel that's made by the adult, the female, who lays the eggs, and then they all go out in different directions from that. And the actual mycelium grows also along in those passageways. So it's a really nice system.
Fab. Well, I think on that final note, we'll probably bring this to a close. So Christian, thank you ever so much for your talk today. It's been absolutely fascinating. I kind of feel I've still got 1,000 more questions that I want to pin you down and ask you. But yeah, thank you ever so much. Thank you everyone for joining. And this has been recorded. It's going to be available on the BMS website very soon. So please share the love. And Christian, once again, thank you very much.
Thank you so much. It's been a real honor and a pleasure to be here tonight. Thank you very much indeed for allowing me to speak. Cheers. And it's been great to get the questions. Thank you so much, everyone, for asking such interesting questions.